how are we going to actually make these trials more efficient? So there's really 4 ways to actually do this, and we'll go through them one at a time. The first is to have a more focused and relevant research question. So the research question we already talked about needs to be one that's worth answering, but focusing it on a more homogeneous population actually allows you to have less variability in the data. So if I only do a study in people over 65, the data is going to be more homogeneous than I do a study that goes from neonates all the way to 95-year-olds. So the other issue here is that it's really important to calculate a sample size after you come up with the research question. Not start off with the sample size and back calculate to the research question. Because what happens when people do that is you end up usually overestimating the effect size, coming up with a sample size that's too small, and you can't really answer the question to begin with. The second thing you could do is change those ingredients of the sample size. Remember I said there's a type 1 and a type 2 error? That is not suggested that you actually do this. But if you allow more error in the study, you will get away with a smaller sample size. But when you think about it, a p-value of 0.05 or a type 1 error of 0.05 means you're wrong 1 in 20 times. If you don't think that's a lot, go on to clinicaltrials.gov and see how many thousands and thousands and thousands of studies there are out there. 1 in 20 is a lot. If you were wrong that many times, we'd be really getting things wrong. The other thing I joke with the fellows is, if you don't think patients care about an error rate that high, just hang a sign on your office door that says, "Guess what? I'm wrong 1 in 20 times," and watch people run out of the waiting room. That is a pretty high level of error. It's also why people usually ask for confirmation of results, because if you get confirmation of results, you're much less likely to be wrong. If you increase the type 2 error rate, you are more likely to discard things that are actually beneficial. So neither one of these is a really good idea. The next thing you can try to do is enhance the effect sizes. Now, you can't make a drug more effective than it is, or hand washing more effective than it is. So what can you do that would actually allow you to legitimately increase effect size? The first thing we talked about is actually try to come up with more homogeneous populations, because that will decrease the variability. You can also try to look for what's called effect modification. Is there some group in whom you think this intervention is more likely to be effective, and then study it in that population? So in other words, like I said, penicillin was actually way more effective in older people who were sicker than it was in younger, healthier people. So you could get away with a study that's way smaller if you studied the effect in older, sicker people. And by the way, answer a more clinically relevant question, because the young, healthy people are going to get better on their own most of the time anyway. So what that does, though, is it affects the generalizability of your results. So if you study a more homogeneous population, you can only draw conclusions about the population that you studied. But this requires what we talked about at the very beginning, understanding the natural history of the disease and evidence from prior trials. So one of the things that really hurts you is enrolling participants in a trial in whom the effect is expected to be 0. So you do a pneumonia trial that has lousy diagnostics, and you enroll a bunch of people with the common cold. You're not going to show anything in those people because they don't have the disease you actually were interested in. So luckily, oncology and cancer drugs have been going this way for some while in terms of developing agents that are more specific to specific types of patients. So for instance, the monoclonal antibody trastuzumab, which is called Herceptin, has been used for breast cancer. It is only effective in people who are HER2 positive and have that specific genetic mutation on their breast cancer, which is only about 20 to 30% of patients with breast cancer. In the other 70 to 80%, it's potentially harmful. It doesn't, and it certainly doesn't help them. So you want to specifically use this in people that are HER2 positive to begin with. So that is studying a more homogeneous population, and you can actually use a smaller sample size, and more importantly, avoid harming people that can't benefit to begin with. Another issue is optimizing exposure. So some preclinical information or data from healthy volunteers may actually help you pick the right dose that you want to study in a future trial going forward. Or if you're doing a study with a drug that may have high variability in the population, you may want to check blood concentrations in the blood. Now that needs to be done by evaluation through a unblinded third party to maintain blinding of the study. And again, you'll hear some more about these various types of interventions and studies when we get more into the course. I'm just, again, giving you a flavor of some of the things that you're going to hear about later. The next kind of thing you might want to do is use continuous instead of dichotomous outcomes. Now, what do we mean by that? Continuous outcome is one where you collect data continuously over time as opposed to just measure it at one point in time. So suppose like what I'm doing in the influenza studies, you want to look at the time it takes people to get over their symptoms from influenza. That's a continuous measure. On the other hand, if I look at how many people are over their symptoms on day 10, when do I measure that? One time, on day 10. So there's not as much information being captured, and therefore I don't have as much ability to detect differences. Now what that does do is it requires more frequent data capture. So what we've been doing now is using patient diaries electronically that people can get to over the internet and fill out at home. So they don't have to be in the clinic to be able to do this. So here's a good example of this. This was a study that was published a decade ago comparing ciprofloxacin to azithromycin in cholera. So notice that more people got better with azithromycin faster than on ciprofloxacin at 120 hours. If I went beyond this, everybody's better in both drugs. So would I be able to detect this difference between the drugs if I looked at how many people were better on day 10? No, I'm going to completely miss it. So this time to event analysis, which is the continuous measure, was able to detect differences that you would not have found if you used a dichotomous yes-no at a specific point in time out here. So the next thing is selection of outcomes. So getting outcomes that are more common actually allows you to detect differences. Now the problem with that is that the more common events may not be the things that you're most interested in. So there's a trade-off here between those things. So sometimes people do what's called composite outcomes, where they look at multiple things that might happen to somebody. So but the problem is that results in issues with interpretation if what you're combining together is not equally important to patients. So for instance, the cardiologists get this right. They use a composite outcomes in cardiology trials of death, stroke, and heart attack. Those are all bad things to happen to you. On the other hand, in oncology, they have an outcome measure called progression-free survival. Progression-free survival means you're alive or dead and your tumor shrinks or it doesn't. They are not the same thing. And if I gave you the choice of do you want your tumor to shrink or do you want to be alive, you'd choose the latter. So the problem is these two things are very disparately different for people. One is really kind of a laboratory radiologic finding, and the other is a direct benefit for patients. That is not like death, stroke, heart attack in cardiology. So, and it also doesn't necessarily imply benefit on all parts of the outcome. You could shrink the tumor, but nobody lives any longer. So, because most of what the outcomes are going to be in the tumor shrinkage piece. Here's what happens sometimes when you combine things together. So this was a hypothetical, this is an article on composite outcome measures by Lupson. And what they did was they came up with a hypothetical trial where the outcome was patients either died or were hospitalized. So if you look at this, it looks like only 5 people in the test group died and were hospitalized, and 15 people in the control group. That's great. So 3 times as many people had bad outcomes in the control group. So this new stuff's great, right? Well, wait a minute. We didn't count the people who died because they never got to the hospital and dropped dead at home. That's the exact opposite. So when we add it all up, there's no difference between the test and the control group. So how you formulate these composite outcomes is really important. You can't leave out things that may be relevant to the patient. So getting hospitalized and dying, yep, that's important, but if you died at home because this stuff was so bad it killed you fast, that's not a good thing either. So you need to think about those ahead of time. So what are some ways to decrease variability? So decrease variability. So we talked about effect size, right? That was moving those curves further apart. On the other hand, decreasing variability is taking the width of that curve and making it narrower. That's the other way we could decrease the sample size of a study. So the first thing is to come up with better measurements. So when you do a measurement, there is the true value and there's always some associated error with that measurement. Well, one way to make that better is decrease the error. And there's 2 types of error. that we already talked about: random error, which occurs by chance alone, and systematic bias. So by coming up with better measurements, you can decrease some of that error, which will decrease the variability in your data. So remember I said earlier about what the problem is when you allow clinician judgment? So this is a really cool study that Archie Cochrane, who the Cochrane collaboration is named after, did in the 1950s. He took a bunch of coal miners in South Africa, and he randomized the coal miners to 4 groups of doctors. And he told the doctors to go ask these people about their respiratory symptoms: cough, sputum production, chest tightness, chest pain, and dyspnea, which means shortness of breath. So these are all the same coal miners. So what we ought to see is the number of symptoms should be flat across A, B, C, D, right? Same people, should be the same amount of symptoms. Look at this variability, right? So these docs got all sorts of different answers to the same patients about whether they had cough, sputum, dyspnea. So, and it also may be how much the particular doctor thinks that thing is important. So Dr. A must love cough, but doesn't think too much about sputum, right? Whereas Dr. C here thinks that, I'm sorry, I got it the opposite way around, right? Dr. C is more concentrating on cough, but less so on sputum, and Dr. B thinks shortness of breath is the more important thing than Dr. D does. This is the same exact measurement being made on the same patients by different people. Because it's vague, because it's left up to judgment, this is all variability. This is what you don't want in your study, because just that kind of variability is going to result in a bigger sample size for nothing, right? You're not actually more accurately measuring what's happening to the patients. You are more inaccurately measuring what's happening to the patients, and it results in you needing to do a bigger study. So coming up with a better outcome measure. So that's why we're trying to develop patient-reported outcome measures that directly ask the patients about their cough, chest pain, tightness, etc., because that will actually decrease the variability by asking patients those questions in a standardized way. This is another great one. So you're thinking, that's all just subjective symptoms. If we use more objective findings, we won't have this problem, right? So Chuck Mortel did this study in the late 1970s where he took marbles and put them between 2 sheets of styrofoam, or this kind of stuff that covers the microphone, squishy stuff. And then he had the doctors measure the size of the marbles. What the doctors didn't know is 2 of the marbles were identical in size. So he was able to measure both inter- and intra-rater variability. And what he showed was that the marbles that were the same size, they had a 25% reduction in their size based on the doctor's measurements. So he was doing this, he was an oncologist, and what he wanted to show was that doctors' judgments about tumor size were pretty variable. So what it showed was that the same marble of the same size got 25% smaller when you left it up to the doctor to judge. That's amazing. That defies the laws of physics, right? So I need these guys to do my taxes for me somehow, that I can, reduce my tax bill by having these dudes look at it and get 25% smaller. So the interesting thing is that reduction is way bigger than the average tumor shrinkage in a trial. So that much noise is really is measurement error alone, it's going to sink your trial to be able to actually do that. So this is and the reason I put this in here is just to show you the percentage of approvals that have used patient-reported outcomes. Now this data is old, it's a decade old now, but what it showed was that some places like pain drugs have been using patient-reported outcomes for a long time, gastrointestinal drugs have been using it for a long time. Here's my part in infectious diseases, 0. So I guess when you have the flu, you don't have any symptoms, right? Of course, you feel fine when you have the flu. So it's kind of crazy that in antivirals and antibacterials, we haven't been using any patient-reported outcomes. Now there's a reason for this, right? When penicillin first came along, it was when people died from infections. But now that we have agents that are actually effective in prolonging life, we need to ask the next set of questions, which is maybe drug X is better than drug Y on decreasing your symptoms, and even though it's not any different in preventing death. So we need to start asking these kinds of questions. So one of the outcome measures that's used often in an attempt to shrink clinical trial sizes is what's called surrogate endpoints. And so these are laboratory tests or physical signs which are used as a direct replacement for how the patient actually feels, functions, or survives. So there are some very good examples of this, like viral load in HIV studies and cholesterol as a substitute for measuring strokes or heart attacks in prevention studies. And those have worked very well. What you never hear about are the surrogate endpoints that completely flop because they don't represent what actually happens to the patient. 2 weeks ago, there was a study in the New England Journal of Medicine that looked at people with Chagas disease, a parasitic infection that you get in South America when you're bitten by this particular kind of insect. What it actually does is it infects your heart, it can give you arrhythmias, and kill you from the heart problems. They took people and gave them a drug called benznidazole, and they looked at whether it decreased arrhythmias and death. Didn't do anything between the benznidazole group and placebo. But the surrogate outcome they used was a PCR for whether you had the parasite in your blood or not. Huge difference on whether you had the parasite in your blood or not. Did it do you any good? Nope, you're still dead. Didn't help you. So, so what's interesting is in my field, you hear people say, that's okay. That's all the drug was supposed to do was kill the bug. So one of my friends who's a statistician said here, yeah, I guess if we threw the patient in a volcano, that would accomplish the same thing. The bug would be dead and the patient would be dead at the same time. So that's the problem with these surrogate outcomes, is that we think that the intervention works only through that mechanism, when in fact there may be unmeasured benefits or unmeasured harms or other pathways of disease that are not measured by the surrogate. So killing off the bug in Chagas disease didn't do anything in terms of decreasing the heart problems. And maybe that's because at that point the damage is already done and it's not related to the bug anymore. It's related to the damage that's happened in the heart, and you need some other treatment for that that is unrelated to killing a bug. So lastly is the issue of assuring follow-up of the enrolled participants in the study. This is a key issue where you spend all this time getting people into the study but don't follow them up. And when you have a lot of missing data in a study, it can really sink your study, because then you don't know what happened to those people at the end of the study. And finally, there are some particular types of study designs, like crossover studies, N=1 studies, and what's called sequential trial designs, that what you're doing here essentially is using the patient as their own control. So in other words, they would get intervention A, then have a washout period, and they would get intervention B. And you compare what happened to that same patient in period A to period B. Now, there are only certain places where these trial designs can actually be used. And usually, these are most useful in chronic illnesses with a stable course of disease. So if you tried to do this in pneumonia, this isn't going to work, right? So pneumonia, you get sick, you're ill, and you either get better or you don't in the space of a couple of weeks to a month. So you can't cross over to some other drug. You're going to be better, or you're not in that short period of time. Some diseases are also worse in certain times of the year. So there's these things called period effects, which can affect these kinds of crossover trials as well. And it's most useful in diseases where the treatment effect is rapid in onset, or there's a rapid cessation of the effect of the intervention when it's stopped. So you have arthritis, you get put on a drug and you get better in a couple of days, they stop it and you get sick again in a couple of days. So those are the kinds of things where this is actually useful. I'm going to skip over this since we're running out of time. The last thing you're going to hear about sometimes in this course is this issue called adaptive trial designs. And really, adaptive trial designs, I always joke, they always remind me of saying, well, I like food. What kind of food? In other words, there's many ways to adapt a trial. So one of the ways it's been used in the past is you study multiple doses of a drug, and at some point during the trial you drop out one of the doses because it's very clear that that particular dose is not effective. Those kinds of studies have been done for a long time. On the other hand, you're moving along and you're doing a study on a particular outcome measure, and you look at the data and you say, oops, doesn't look like I'm going to have much of an effect on that. I'm going to change horses and switch to some different outcome that's not as relevant to patients. Well, that's a lot different. You've adapted the study in both those situations, but one of them is legitimate and one of them is not. So you'll hear about this in terms of advantages and disadvantages. I am going to make a caveat on this final slide here, and that is almost everything I've talked about today in terms of modifying studies to make them more efficient is in the setting of trying to show that one intervention is better than another one. That's a superiority hypothesis. There is this crazy thing I mentioned earlier called non-inferiority, which is a misnomer. Now, to you and me, non-inferior means not worse, right? At least that's what I was taught in English class. But this trial design is actually designed to rule out that the new intervention is worse by some clinically acceptable amount. Now, there are places where this makes sense, and the reason for these kinds of trials is there are places where patients may accept somewhat less effectiveness for some other benefit like decreased side effects. So for instance, uncomplicated urinary tract infections in young, healthy women. Suppose we come along with a new drug that profoundly causes less nausea than an old antibiotic. So a woman might say, yeah, I'll take the drug that's a little less effective in curing my UTI symptoms because I don't want to be throwing up all day. On the other hand, when the outcome measure is death, like somebody in a ventilator with pneumonia in the ICU, how much death is clinically acceptable decrease in effectiveness? The answer to that is 0, right? Nobody wants to die. And in fact, you'd accept a drug with more side effects if it kept you alive, right? So there are places where this kind of hypothesis makes sense, and there are places where it does not. So you'll hear some more about what are some of the issues in designing these kinds of studies. And in fact, some people have held that in certain situations, these trials are unethical because they disregard patient interests. One, because they shouldn't be used in life-threatening illness, and two, because that other benefit of, say, decreased side effects or improved convenience is never spelled out for the patients. So the study's designed to show new drug is a little less effective, but it's never evaluated, okay, so what's the benefit on the other side of that equation, like decreased side effects? If that's not evaluated, then all you've done is put patients at increased harm without being able to tell them they're getting something else on the other side to balance out that equation. So in conclusion, developing an efficient trial starts with planning and a good research question, and then some more planning, and then some more planning. So I just want to get across to you that that's the real key to this, is really thinking about these things ahead of time. The question comes first, and the sample size comes second, not the other way around. And there are various methods to increase effect sizes and decrease variability, which when applied in the correct setting can provide valid and reliable answers to your research question. And for some diseases, developing the tools is a good way to start. For instance, developing better outcome measures and having better data on the natural history before you get to the point of where you want to do clinical trials.